screen. Great. All right. Participants can see, you can see. And what I'd like you to see is sort of a broad spectrum of the work that we're doing, just to sort of provide some greater context for the things that we do in our group. So while a lot of the work, basically all the work I'm going to tell you lives within this phase, the type of things that we do and the reason that we're interested in problems related to sequence composition and architecture design are often motivated by things in these various types of thematic areas that underlie the research problems that we choose to work on. And so thematically, we're very interested in the way in which macromolecules can respond to different types of stimuli in efforts to potentially control them, maybe via sequence or composition type of design. We want to understand sort of the underlying physics that drive that phenomena at the same time. So a lot of what we do is a sort of a balance of both sort of the applied machine learning stuff as well as fundamental mechanistic inquiry. In that way, we're very interested in these types of messy systems, heterogeneous type of media, and how that influences the properties of materials. So we'll think a lot about the presence of interfaces and how that might modulate the behavior, as well as how we can design or think about the interactions of systems that live in this sort of like biological interface, either inspired by things that we see in biology or maybe synthetic things that we intend to interface with biological media. Now, to continue along these lines, this is to give you another sense of the type of problems that we think about. So we construct theories and different types of models to try to describe the phase behavior of systems. That way we can think about that stimuli response sort of problem. In the messy sort of systems, this heterogeneous media, we think a lot about interfacial properties, ideas creating coatings or adhesives, things of that nature. What in the composite space dispersions, where you have these mixed type of systems, that's going to influence the way in which they behave. And at the heart of all of this type of work, we're thinking about what are the implications of chemistry, right? And how we can use that to control or understand emergent phenomena or properties of those types of materials. But also, to continue with sort of the theme, there's a through line here in terms of our interests that are not entirely focused, but very prominently featured in the way of polymeric materials, right? And that's because when you consider polymeric materials and the properties, the phenomena that underlie their function, the chemistry is incredibly important, right? That's at the heart of everything. But it's not just the chemistry, right? It's also the way in which you arrange those types of constitutional units, the composition, how much of them you have, and even the architecture and other types of aspects, right? The methods of processing, you can get very different materials properties from the same underlying chemistry just by the way that it's processed, right? And so this is sort of a distinct challenge, and it's an interesting challenge that distinguishes polymeric materials from a lot of efforts in, say, more small molecule space, is that all of this complexity that we have, can we unblock it to achieve interesting things in the way of functional materials, have you? And it also is sort of a reason, all these factors, as to why you have such prolific influence in everyday life. And so it invites many questions, right? Like thinking towards the future, yes, we can make some of these things now, but what could we do if we had very fine control and understanding of all of these factors and what that means, the implications for the resulting materials properties? And we'd like to therefore think about the following questions that I pose at the bottom here, which is like all these factors, what it amounts to is going beyond just chemistry. There's so many knobs that we can turn, right? Constitutes a vast design space, and we're often in search of a specific type of thing or optimizing a given figure of merit. So how do we find the right thing? In light of the fact that a lot of the methods that we might commonly think about may not be immediately appropriate for addressing the complexities induced by macromolecular materials. And ultimately, how can we navigate design? And we obviously think that machine learning has a large role to play in that process. So I'm going to give you a little bit of perspective. This slide, I'm going to try to teach you, not really teach you, I'm going to show you, right, a timeline of developments in machine learning, because we have sort of a recency bias as to what has happened recently. That makes this seem like a very new field, if you will. So here I have a timeline with some recent things that you're quite familiar with. Over here, right, we're here today in 2024. There's recent stuff that's happened that we're commonly using and deploying. And there's been a lot of interest, right, in utilizing machine learning techniques in physical science and engineering now that is continuing to grow. So if we think back, and we'll begin at sort of the Big Bang, then lots of stuff happens. And then we can kind of chart forward, right? And so while I skipped over a lot of stuff involving dinosaurs and these types of things, you can see that actually like the earliest proposition of things that resembled neural networks go back to the 1940s. So the theory, right, for what constitutes even modern day neural networks was proposed quite long ago. And this was kind of coincident with early developments in computing devices. So here's a fellow that probably many folks nowadays recognize more commonly. We have Oppenheimer here, joined by von Neumann. This is at the Institute of Advanced Study, which is kind of adjacent to Princeton, and they're talking in front of one of the early computing devices. And these are just like a series of sort of landmark things that happened along the way, which you can see has occurred over many decades, despite sort of the recency of what we understand machine learning to be. And actually, interesting for me, coming from somebody who, thinks about polymer physics and the like, a lot of these developments are very contemporaneous with, major landmark papers and discoveries in the polymer field. So they're actually relatively similar in terms of age in that regard, but it took a while to get traction. Another thing that I like to point out here is in 1972, so I'll point out this Whitehill report, okay? And this was sort of a review of the field. We're very familiar with these things today. And this is an excerpt from that report where Whitehill remarks that, guys, we've had machine learning since 1950 and nothing has come of it, right? And so it's kind of remarkable that this hype existed then, and we see it again today. But in all honesty, Whitehill may not have been wrong, right? It was kind of, I don't imagine that this made him incredibly popular amongst colleagues that were looking for funding and that sort of thing, and actually spawned this era of AI winter. But what we were really waiting for, right, was for the right advances in computing. But the point is that this has been around for a long time. And we can nowadays see that, right? So now machine learning is sort of pervading all manner of material science to varying levels of degrees, right? But it's changing the way in which we think about studying systems. And what that is quite emblematic of, right, is an evolution, an augmentation of paradigms of scientific discovery. And so you may be familiar with some of these terms, wherein back in the early 2000s, Microsoft Research said, hey, this is a fourth paradigm of scientific discovery, which is to be based off of not data-driven science. People always say, all science is data-driven. This is true, but the presence of big data, right? Available data. What can existing data help us to understand about the world today, given that we've produced it? And this was sort of the notion of the fourth paradigm science. Like, can we create databases to show us something interesting, to find materials that are working within the stuff that we already have measured? Can we learn the correlations, the relationship from chemical to structure to property? Does that exist? And that was the fourth paradigm, right? Doesn't replace the other paradigms, but it's another thing. It's another option. And a couple of years ago, we sort of announced the fifth paradigm of scientific discovery. You read this article, it does a terrible job of describing what the fifth paradigm of scientific discovery is, but I'll tell you what I believe it amounts to. This is now not relying on the landscape of available data, but utilizing machine learning as a way to guide our data acquisition. So it's bringing in elements of AI and machine learning to help us know what data to collect on task. So this is in the vein of like Bayesian optimization, active learning, materials discovery type of stuff. So that's the fifth paradigm. I checked this morning. I check every morning just to see. I'm here to suggest that there is a sixth paradigm of scientific discovery. Again, these are augmentations. They don't replace. I want to get credit before Microsoft does. So sixth paradigm of scientific discovery. I think there's been sort of a phase change with developments in large language models, these foundation models that are also heavily influencing the way in which we think about scientific pursuits. And so I would say that that's already here, and we're continuing to sort of flesh that out. But I'm going to show you today sort of what that has meant for us over the last couple of years of research in our group. And I'm going to endeavor to tell you a few vignettes, okay, that exemplify the ways that we've utilized machine learning. Primarily, these are, I would argue, mostly fifth paradigm type of stuff if you want to play it in that way or characterize in that fashion, but with a vignette about sequence design, composition design, and then architecture. And of course, because sometimes I get distracted by what I'm saying, I don't know if I'll get through everything. I want to be sure to acknowledge the students, postdocs, and collaborators who have heavily influenced, contributed to the work, and the various funding sources for these different things. So let's start ahead with the story about sequence design. So a lot of what we do is we're interested in sequence design. We need to find some inspiration for the problems that we work on. And in this particular vignette, the inspiration comes from the field of protein condensates, okay? Which, if you don't know anything about condensates, is fine. I hardly do either. But I'll give you a brief primer, okay? So these are bodies that form sort of spontaneously in the cell that are very important and critical to a lot of cell function and physiology. Okay, so they underlie the formation of all these sort of membraneless organelles, the function of which is to roughly control like who's in, who's out, who's good, who's bad, and basically facilitates a lot of the things that need to be done within the cell, transport of cargo, getting rid of waste, all that kind of thing. Now, the interesting thing about them, at least a subset of them, is that the formation, the spontaneous formation, is actually underpinned by thermodynamics and critical components often being proteins, intrinsically disordered regions of proteins, other biopolymers, of course, okay? With the notion being that depending on who's there and who's present, these different components, you can achieve a lower free energy state by spontaneously phase separating into sort of a biopolymer rich region. That would be kind of like this right side of the room, and then a biopolymer lean region, which would be like that side of the room, okay? So, and the condensates themselves, you can see them through this microscopy, that's the biopolymer rich region. Okay, so they're important for many things, cell physiology. I'm actually gonna take the view that these are intriguing stimuli responsive biomaterials, okay, because they trigger and they form in response to some stimulus, and then the functions that they perform depend on the material's properties. So here is a paper from some Princeton colleagues and other collaborators. I was not involved, so I just read the paper, found it interesting. And these are binodal envelopes illustrating the phase behavior for a range of different systems, different proteins and RNAs, not super important. What the interesting thing for me of the paper is not really the point of the paper, but this plot here shows the measurement of materials property, not often done. Usually people are very interested in the phase behavior of these things and what drives the phase separation, but less so measurement of materials properties, which is hard. So here they look at the viscosity of the condensates that are formed, and they're plotting it against this metric on the bottom. There's a second virial coefficient multiplied by a concentration. This is a proxy for the propensity to phase separate, okay? It can correlate with things like the height, like the critical, in this case, critical concentration of salt that induces it, or the temperature, critical temperature. This was sort of like a pseudo temperature axis, if you like. And so you see this type of correlation, which I'll roughly characterize as being that the more that you want to phase separate, the more sluggish are the dynamics within the condensed phase, the higher is the viscosity. So this is something that can be kind of understood from physical principles. If you like, the second virial coefficient, it describes these interactions between the different components. And if you have a very negative second virial coefficient, then you like each other components quite a lot. And so you might imagine that they would stick together, maybe cause some additional friction, something along those lines. You could imagine how that would lead to a more sluggish condensed phase. Whereas if you have a positive second virial coefficient, that means you don't want to interact with them, you'll run away. You don't coalesce to form, a very condensed phase, sluggish environment. And in fact, these are simulations, okay, that we've run to illustrate this principle for homopeptides. So these are homopolymers just consisting of a single type of amino acid or residue, where using coarse-grained simulations, where again, we're plotting some proxy for propensity for phase separation dependent on the second virial coefficient. And then on this axis, we have not the viscosity, but the diffusion coefficient of the protein, the homopeptide, in the condensed phase. So we found a condensed phase, and you can see that they align. And we can actually figure a reasonable theory to show that this should be the case for a homopeptide, because there's only really a single energy scale that's available for this to happen. But in the peptide space, there are more things that we can do. It's not just a single amino acid, but we have 20 of them, and we can arrange them in different ways. And so we endeavor to know that, suppose that most things look like this, okay? Can we perform a search, which is going to be guided by this sort of framework shown on the right, where we want to see how far we can get to find sequences, protein sequences that simultaneously, for example, are the fastest that they can go, yet have the strongest propensity for phase separation. And if we do that, then sort of this dynamic range between whatever we find here and what's down there, that's sort of a region of tunability, if you like, where we could have stuff that phase separates at the same temperature approximately, but exhibits a different underlying materials property. This is sort of the rationale. And so the way that we're going to approach that is a common strategy that we use in my group, which is that we do high throughput molecular simulations, often with coarse-grained modeling, where the objective is not really to measure the final figure of merit itself, but we have to find things that are cheap because we're going to do lots of them. And so we have some set of proxy calculations that get at the heart of the physics that we think is important for the figure of merit. So here we calculate second virial coefficients. We map out an approximate equation of state. So what is the pressure as a function of density? This gives us some sense as to whether things will phase separate or not by trying to identify a van der Waals loop. And for those that do, we can measure with short simulations, get an estimate of the dynamics in the condensed phase. Then once we have that data, we will train machine learning models, okay? The details of which are not super important, but what's interesting for this task is we simultaneously would need to figure out how to ensure that we find things that do phase separate. So we have a classification task to examine whether for this given sequence, will it form a condensed phase or not? If it doesn't, you don't pass go, you don't collect $200, we just move on, okay? But if it does, then we'd also like to know what are the estimated diffusion coefficient in that condensed phase. And so we build a Gaussian process regressor model to estimate these quantities and a classifier to determine this other thing. And those get rigged up to an optimization task where we can characterize this in the search for a Pareto front, which is the sort of boundary line of trade-off of these materials properties, okay? And so there is an acquisition function that exists, this expected hypervolume improvement, which helps us select the next set of sequences that we think will measurably improve on an existing Pareto front. And that's the way that we tackle this active learning task. So this is everything that I just explained, okay? Now, I'll say that in part, the success of these efforts, the utilization of machine learning depends on us being able to fairly represent or describe these sequence-defined macromolecules. This is a problem that we've thought quite a bit about, okay? And we discuss heavily in this paper. I'm not going to tell you or go through the details of that work, but reference it and just deliver you one piece of guidance, which is that what we find in these sort of like data scarce regimes, which is often what plagues polymeric materials, that simpler representations are better. And so what we do is we describe the polymers not explicitly with the sequence, peptides explicitly with the sequence, but we have the representation of what is the composition, the relative like mole fractions of incorporation of all the different amino acids. And then what we have is some derived set of sequence descriptors, which are commonly found elsewhere within the literature. And these are things that come from the sequence. So we know what the sequence is, we're just distilling the information. And this works a lot better, we find, than trying to learn from the sequence itself with some of the more, we'll say, like fancier feature extraction architectures that are out there. Okay, so here's kind of the final result, all right, post about 2,000, 2,100 sequences, simulations that we've run. Actually, many more simulations than that, because every sequence requires many different kinds of simulations, but 2,100 sequences characterized. And this is the expression of our dynamics. So we have more sluggish dynamics down here, and then things that like to phase separate more oriented along this axis. And this is our identified Pareto front, all the starred materials there, okay? We don't explicitly show all the other sequences, just sort of the density of sequences in these various regions. The original data that I showed you for the homopeptides are the open white circles. And so once again, just to drive home, the stars here, the Pareto front, what these represent are sequences for which we cannot find any other sequence in this space that measurably improves simultaneously both of these quantities, okay? You could find something that's around the same, but not a game changer, not a major delta, okay? And this is sort of like, I guess, expressed if you look at our progression towards completion, where we're just not finding anything new after 6, 7 iterations. We consider this as more or less a converged version of the Pareto front. And once again, this highlights, it gives us a sense in the context of this model, okay, of what is the tunability that we're looking at least in one direction, okay, where the dynamic range, so say the distance between this point and that point, if I were to draw a vertical line in that direction, so the vertical line roughly correlates with like the critical temperature, so when it would start to form a condensed phase, some measure like that. And then this is sort of the range of tunability and the dynamics. So you get maybe a little bit more than a factor of 2 in certain regions. Down here, there's very little wiggle room. Everything is just very, very slow, and it's hard to make them go fast if they want to come together so strongly. Now, did we get anything from this? Ultimately, now the machine learning, the models that we train, those are free, but there's lots of data that goes into it. So when we figure in all the data that was required to train the models, and then compare that to what would have happened for a set of validation tasks. So here what we're doing is sort of much more expensive simulations that are on the order of like 100,000 particles or so, where we're actually not looking at proxies of the formation of condensed phases, but actually observing what they look like and measuring the dynamics within here. We find very good correlation with these expensive simulations and the results that are proffered from machine learning on both the phase separation propensity and the dynamics. So that means that models did a good job overall. But ultimately is about a couple of order of magnitude faster than if we had conducted the same search, but using these types of simulations instead of the proxy type of calculations that we crafted. So this is more like a validation that the proxy calculations that we invented to try to generate enough data, were they effective were they worth it? This is the extent to which that's the case. Now, the other thing that we try to do, of course, is we have all this data, and yes, we've solved the problem that we set out in terms of like identifying this Pareto front. What is the trade-off line? Can we learn anything more from the data that we've generated? And one method that we're working with to do that is in the field of this like interpretive AI, explainable AI sort of stuff, in this idea of a counterfactual analysis. And so the notion of what we're doing here is we have like two sets of things. We have things that are Pareto optimal that live on the front, and then we have things that aren't. So you have to define, say, two classes. And then when you do that, what we'll search for is for every given thing that lives on the Pareto front, we'll look at all the sequences that are in the other class, so the things that are not on the Pareto front, by a measurable threshold. And we want to maximize the similarity between those sequences, okay? So whatever is the residual difference between those sequences serves as some level of possible attribution for why something is on the Pareto front versus not. It's kind of like if you were going to go buy a car and you're looking at a lot of features of cars and they all have about the same price, they hopefully all have four wheels, like those things would not explain why you preferred one car versus the other, but one may have some other feature that we could look, irrespective of whether I talk to you about your decision to buy a car, we can just contrast the features and they represent them and say, aha, well, these things are all the same, but for this couple of things. And so we may surmise that that's the reason you bought that particular car. So this is the counterfactual analysis performed for different features. We have like the degree of polymerization or the sequence length, some notions of hydrophobicity, patterning of hydrophobicity, charge, and this sort of stuff. These things are messy and complicated. The distillation of this, if I had long enough to tell you about it, is that there are roughly three different regimes that we find here. This one, and this one. And we find through this counterfactual analysis that, say, for example, in this region, where you have, I guess, more positive, more positive, closer to positive second virial coefficients, faster diffusing. These are dominated by polyamphiolites, actually, which is interesting. Whereas down here, the way to make things really strongly face upright and slow is usually by incorporating large amounts of very hydrophobic residues and heavy ones as well. So a lot of the sequences down here contain like large amounts of tryptophan. So the point here is that we can actually learn something from the data that we generate afterwards that goes back to some sort of physical principles that might be, roughly used as design guidance or principles that we're kind of exploring further. All right. So now I'll tell you about different type of tasks, very similar, but in the space of that leverages composition design. And so the inspiration for this relates to really like enzyme catalysis and the numerous types of applications for which it may be used. So enzymes, of course, now these are like structured proteins. Prior vignette is unstructured, disordered stuff only. But they can be very highly selective catalysts. They can have good activity. And so they're desirable for a lot of different industrial processes. But the maybe crux of the problem in certain circumstances is that they may not work, right? If they're evolved to work in some scenario, we want to expose them to some sort of like harsh set of conditions. They may fail. They don't work. So we want to kind of improve that. And one of the strategies, which was sort of exemplified in this science work from Ting Shu's group, showed that you could actually use random synthetic homopolymers of some set of composition that in some way interact with enzymes. This is horseradish peroxidase. So in the presence of these polymers, the enzyme is able to survive in a harsh environment. And this is illustrated on this figure of merit here, which is the percentage of the native enzyme activity, which is retained in this new thing. So a regular old enzyme in its happy state is 100%. But the difference here is that they're looking at this enzyme in toluene, okay, which is a very harsh solvent. Ordinarily, in the presence of nothing, it would denature that enzyme quite readily. But this is a comparison of different stabilization strategies, this being the random heteropolymer strategy, composition design in a complicated way, I won't get into, but it works pretty well. It survives in toluene for a measurable amount of time. These are sort of more chemical agnostic type of approaches, micellar encapsulation, they work less well. And so this is quite promising. So we can kind of ask the question, let's imagine that we have the capability to build polymers and then put them in the presence of these proteins. Maybe they form these protein polymer hybrids. Can we effectively design those copolymers that effectively stabilize any given enzyme that we're granted? And so we've endeavored to study that problem first with the proof of principle with like 3 sort of canonical enzymes. Again, horseradish peroxidase, glucose oxidase, lipase. This is sort of the chemical toolbox that we're working with. So all the copolymers are formed from these monomers here in different fractions of incorporation, different lengths. So those are the knobs that we're turning to try to optimize the activity. But we're not going to use simulations. I don't simulate a lot of proteins. They sort of scare me. To do something that would relate to this figure of merit is sort of like beyond our current modeling capabilities, I think, certainly beyond mine. But luckily, we don't need to. So this is the lab of my collaborator, Adam Gormley. His student, Matt Tomasi, is very critical to the work. And you're seeing sort of an automated robotic platform for polymer synthesis. And here it's just mixing things. So it's grabbing monomers, it's putting them into different 96 well plates, and it will initiate the polymerization for a given amount of time based on light. And that's how they can roughly achieve, these prescriptions here. The only limitation, a limitation that we impose is that the copolymers we make, it has up to 4. We can choose up to 4 of these things. And that's because that's how many stuff the robot can grab at a time. But otherwise, we have a very similar workflow as I showed you for the biomolecular condensate thing. You see a lot of these workflows, of course. And so, initially we have some data set of polymers that the Gormley group routinely makes. And so that's sort of like a human crafted data set that they think gives them a good sense as to what's going on over the chemical space. We could debate that. And then what we can do is they take their data. So the important thing is that they can actually do these assays, the enzyme assays. This is a hard part in reasonable throughput, okay? And we can take that data, build machine learning models, perform the same sort of optimization where our figure of merit here is, can it optimize that retained enzyme activity? Now, by contrast to the work from Ting Xu, just to make sure I say, the type of adverse exposure we're looking at here is thermal denaturation instead. So everything gets heated up to a high temperature that would denature the protein in the presence of nothing else. But now we have the polymer there. Things do go back to room temperature. That's where the measurements are made. Okay. So here's the result of several rounds of optimization. I think all told, for every enzyme, maybe 600 polymers characterized or so, ballpark. Do we need that much? That's an open question. But what we're showing here, I have a penchant for making very complicated plots, so I'll show you what's happening. So this is our figure of merit. These 3 things are the 3 different enzymes, okay? And all the markers are measured data points. So this is all measured data that has been collected after the fact. And we're showing the distribution as a function of the degree of polymerization or binning them in those categories, just for some. Now, I explain all that. Oh, and the coloration corresponds to some rough interpretation of the chemistry of that copolymer. I tell you all that, and then I'm going to tell you to ignore it, because what we're going to do is we're going to histogram the data upwards based off of when the data was collected. Okay, and then we get these distributions, which are shown up top. So the blue distribution is where we started with the initial like handcrafted data set. And for the most part, all that data is clustered towards this side, which is to say that they're bad, right? Then the orange is sort of our progression, collecting overall data, the next couple of rounds of active learning. I think iterations 1 through 4, we're collecting all of that data, binning it that way. The vertical lines, that indicates the mean over all that stuff, just to visually understand that we moved right, okay, and by a measurable amount. And then the green is sort of a final exploitation round, where we just say, give us the best things that you think you can give us. And that's the right distribution, the right most, the green, where for each 3 enzyme, we found a set, not just one, but we've found a set of copolymers that actually do the job reasonably well. So once again, we're going to try to understand a little bit about what we did here, and at least get some insight as to what makes these tasks different and whatnot. And so this sort of spider plot here is showing the average features, okay, that define each of the copolymers. So this is degree of polymerization, sort of the average length of this stuff. And then the rest of this, these are the monomers that I showed you while introducing the problem. And the main takeaway from this, with each of these different webs that are formed that correspond to the different enzymes, is that they're different shapes, which is to say that each of the enzymes have sort of unique preferences towards the copolymers in terms of what results in an effective stabilizing composition. So this is like effectively, proves this hypothesis that tailoring is an effective strategy, say. We can also sort of examine to what extent, now these are the things that are found in the top, the top 10 copolymers for each. We can also understand, to what extent are each of those features important for driving the stabilization. So it's not telling us how, it's just telling us which ones are or are not. And so from this, these are organized from top to bottom in terms of like what's most important versus what's least important. And then when you see red dots, that means that it's a high feature value. So red here would mean longer degree of polymerization. And so for this, we can see for glucose oxidase, I suppose the interpretation is that having longer polymers is actually detrimental to your figure of merit, which this shows, that determines like where you are horizontally. And so we can sort of organize those things. I think it's interesting what we can find from this type of analysis, in the sense that like for certain polymer or certain enzymes, degree of polymerization is an important factor, whereas in other ones, it's relatively low on the list. So that's something that we can, further investigate to try to understand. And once again, there are other sort of like subtle distinctions where we take, say, two monomers that appear relatively similar. So methyl methacrylate, MMA, butyl methyl acrylate. Why is one preferred versus the other? These things we don't really understand, right? But this points us to where we might want to look to understand it further. And then finally, we just showcase, in this case, this is mostly like an experimental study, but it illustrates that the polymers that are designed, they're rather specific for their task. So this makes sense. I mean, they were tailored for the given enzyme, but just what we didn't look at is, say, they were in the presence of other enzymes. This shows that the ones that they're designed to work with, the enzymes that the polymers are designed to work with, are the ones that they end up stabilizing, right? Whereas the other ones, they don't really impact the performance that much. So we can also get some kind of interesting insights into the mechanism, or by finding interesting materials, it gives us something to chew on. And so this is more experimental data, circular dichroism, which the sense of which we're showing 3 different conditions. And this is, again, some data collected from the Gormley lab, where at the bottom, the black lines, these are sort of unstressed state, looking at something about the protein structure, okay? And so everything lives down here. Then the idea is that when you heat it up, everything moves in this direction. You get to the blue curves. And the interpretation of this data is that actually, for this particular case, the enzyme's still unfolded. Okay, that's sort of like the dissipation of some of the features here indicate that the protein's still unfolded. But then when it was cooled back down, you can see, say, the solid line, which indicates the protein in the presence of the polymer, goes back measurably closer to the initial black line, which is to suggest that it's actually likely helping it refold, okay, not preventing it from unfolding ever in the first place. So if you don't understand circular dichroism, and I don't, this is roughly like kind of the idea that things unfolded and we helped it refold. And this is sort of highlights a new possible mechanism. It was interesting that we all believed within this field that the polymers were coming in and sort of like hugging and protecting the protein, which may well happen in many circumstances, but in this case, not so. I'll go ahead and skip this. So another thing that I'll point out about this problem, which is interesting, is I show you how this all works, right? It seems like it works quite well, but sometimes there are things that we fail to account for that are maybe pervasive in other problems. So here I'm showing you a heat map projected onto this sort of more intuitive chemical space, which is supposed to show you how we traverse this design task. So the stars, if you can see them, indicate the average characteristics of all things that are proffered within different stages of the active learning, which is distinguished by the color. Okay. So in the background, the heat is after the fact knowledge of roughly where the promised land is. So things that work well are indicated by the yellow. So, all right. So 2 different polymers. This is lipase. If we start nearby to red, okay, which is the first iteration, this is a simplification of all the data, but it appears as though we just sort of like run over to the promised land. We march readily just there, right? So it was pretty easy to navigate, you might say. For this enzyme, glucose oxidase, you start at red. And then the next step, you find sort of a very interesting region here, which is orange. And then it reverses course. So the reason it reversed course is because when it moved in this direction, which is incorporating more and more hydrophobic materials, what happened is Gorman Lab came to us and said, hey, everything is gelling or everything is aggregating, and we don't want to work with that kind of stuff. So we had to sort of on the fly incorporate a classifier that would dissuade propositions in areas that are likely to engage in this sort of like undesirable physical characteristics, just because it makes it more difficult to measure. We don't know what's in there. And so that's what we did, sort of on the fly. But this is actually kind of a pervasive problem that occurs in many kinds of design tasks. And so it got us thinking, like, rather than building this thing on the fly, can we learn how to more efficiently classify materials behavior in general? And then they can just be hooked up to different type of design tasks. And the notion here is that there are many strategies for different types of data collection. And so here's just an illustration of that, of like what constitutes a data selection strategy on this Princeton P data set that we invent. So a strategy consists of how do we initially allocate the data that we begin with, and then how do we subsequently acquire new data, and then also in what ways are we modeling the underlying function to describe this thing. And so so here you can see sort of the evolution of two different ways using different machine learning algorithms, different initialization, and so on. So ultimately, this is a robust study, I'll say, where we look at 40 different strategies, 25 different tests. Here are just two that have something to do with polymers. Many of them are more, other elements of material science that we could pull from the literature. So this is a combination of data that we've generated over the years, as well as other data sets that we think roughly thread the needle as to what is a relevant classification task that we might care about. And so these are the different strategies. The definition of strategy, this is roughly speaking a figure of merit that quantifies how well it does. And the color is just a quick visual interpretation as to the underlying modeling, the machine learning algorithm, okay? So two different things. This is polymer solubility. This is something to distinguish whether it'll be a coil or a globule. Main takeaway here is, we'll say on this side, we can see red clusters up to the top. So tree-based algorithms, these random forest things, gradient boosted trees, those are the top performers. On this side, neural networks appear to be ones that are a little bit more well-suited to the job. I previously held the opinion that the actual algorithm itself is relatively unimportant. But here we see kind of that actually there is some distinction in terms of what's happening in the way that these things progress, and it's different, right? So what are we going to do with that? I mean, it's sort of unsatisfying to have a conclusion, which is like, well, things depend. I mean, often that is where we get to, but can we at least learn maybe when things work better versus not? That's one way to go. Or we do something a little bit different. And so here what we thought about trying at least is instituting a concept that's like roughly in the vein of meta learning, where we're going to try to just build models where if all these algorithms work well in some cases versus others, based off the data and while we're acquiring it, can we leverage information across different models and combine them in ways and basically learn to give more opinions or weights to ones that seem more well-suited as a function of data collection. So we're actually not going to try to find which model works best. We're just going to aggregate all the models into an ensemble and then actually have learnable weights as a function of data acquisition that will give us our final prediction. Okay, so this is coalescing all the data across all the 40 tasks. So before the figure of merit was for a single task, here we have a measure where we're still trying to figure out the best way to quantify it, but it's like, what fraction of tasks is this the best, right? Or to what extent is it the best? Or how does it compare to the best algorithm? Okay, something along these lines. It's better to be more towards the right. And I guess nicely, what we find is that if we just take this ensemble approach, which is characterized by the orange bars, those seem to be the ones that gravitate to the top. So going forward, I think the idea is that, well, if you just want a reasonable strategy, knowing nothing else, right, this seems like a relatively simple framework that you can implement to guide your data acquisition that is, just something that can be a drop-in until maybe you have a motivating reason to do otherwise. But if you don't have any motivated reason to choose one versus the other, this seems like a fair way to go. Okay, so I'm just about, I'm running out of time here. So I'm going to briefly walk through, I'm not going to walk through, I'm going to run through just the idea of architectural complexity and just hit on some of the high points, I guess. In the first two problems, what I'll say about them is that while the design spaces are large, at least I can define them. So with a pen and paper, I can sort of write down the number of unique things that I think comprise the space. If we introduce architectural complexity, this becomes sort of a nightmarish problem, to where the typical ways in which we were solving the optimization in this case no longer works. And then if we think one step further, not just architecture, but if these things then begin patterning, it's just completely out the door. Okay, so we have to do something different. And this is where we've leaned on a combination of things that we've learned for like linear homopolymer or linear sequence and composition defined type polymers, but now incorporate elements of generative modeling to describe these complex polymers that have interesting architectures and the ways in which we're going to do that. So we still say feed in a set of descriptors that are like definable from the architecture itself, just in the same way that we found was useful for the sequence. But then we also directly feed in information about the architecture. And then all of this is processed in a variational autoencoder framework to give us a low-dimensional representation that we can learn from. We can do the machine learning on as features from that space. And we find that this works very well. It gives us a physically interpretable low-dimensional space when it's coupled to different learning tasks. So you can see that here, where these are like projections of coordinates in our low-dimensional representation of these different architectures. And so they kind of nicely organize with respect to what is sort of more collapsed. So this is radius of gyration coloration. And then it also, sensibly organizes architectures. Okay, so we can have an understanding as to how those relate to each other. Linear polymers are obviously simple, but when it comes down to distinguishing a branch versus comb versus star, where do these live within that space? And so this data-driven framework does that. One thing that we're doing with this is we can now design new studies where we control for different variables. So for example, here, we're interested in sort of the rheological behavior and the implications of architecture. But this is sometimes quite hard to decouple from other variables like just the polymer size, because at like low concentrations, everything is just going to be typified by the average size of the polymer in the solution. And so either you have to do a thing where you are just systematically sweeping over aspects of a given architecture, but other things are changing. So what's your level of attribution as to what's the effect of the topology? And so here we can say proffer a set of different architectures, different manifestations that live in these different classes, and they all have the same size in dilute solution. And then we can study their rheological behavior and gain some insights from that. Now, finally, just the last thing I'll point out as a jumping off point is like that only solves a part of the problem with architecture. And even in that data set, we're relatively confined over certain things that are important, like the molecular weight range. And so we want to learn, how can we generalize our models, not just over topology, but also how large they are, make them more useful in that regard, as well as patterning. What are we going to do with that? So this is just blowing up the space to where we can't feasibly collect data over everything, right? So we need to create models that are better at working from limited amounts of data, sort of cool. And the way in which we're doing that we've had this idea for a long time and it was just never that useful until now, is we can sort of craft the prediction task to not try to do everything, but rather we can formulate from this sort of a baseline model, which is predicated off of maybe a polymer physics theory or something. And then the role of the machine learning architecture is to learn the deviation of the theory to sort of the final property, okay? With the expectation being that providing this theory, which polymer physics is very good at, say, for example, scaling and that sort of thing, that can handle some fraction of the complexity. And then this is just left to learn the balance, which may be a simpler task. And so this is a brief comparison to show that this works in terms of generalization across different degrees of polymerization. So here we're training on small polymers, and then we're testing on large ones. This one, we're training on homopolymers, and we're testing on copolymers. So we're looking at extrapolation type tasks, if you will. And this is how the machine learning does by itself. It sucks. If we look at just the polymer physics theory, it's also, maybe not fantastic, but I'll leave that to you to adjudicate. But this is sort of our tandem, one of the tandem architectures that we've created, and now we get some reasonable predictive capabilities. So we're really excited about this, and I think it'll be a promising thing to move forward. But it's really only just, a component as to what we're working on. So everything I told you today, or what needs to be worked on, everything I told you about precisely defined systems, simply defined and in single conditions, there's a whole host of things that work beneath the surface, which are going to be needed innovations in the future to really get these things towards representing new polymeric and real polymer systems. So with that, I'll leave up these types of conclusions. If you can read them really quickly and quickly acknowledge my group again, some of the references, and I'm happy to take any questions.
Hey, questions, Ken?
So on the second one with the enzyme, yeah, my initial guess was the things that work are copolymers that stop unfolding.
Mm-hmm.
But what you said you learned was they don't stop unfolding. So presumably something happens that goes along that reaction toward unfolding,
Mm-hmm.
but they bring the system back to the folded state.
Yeah.
So can you give me some idea like how far along in the typical case they are in this unfolded state before, the copolymer does its job and brings it back?
I'm not really sure, to tell you the truth. The nature of the assay is it's like immediately subjected to the thermal exposure. So it's not like they're ramping up the temperature slowly to see when things happen versus not. I don't know if that's what you're getting at.
Well, I'm saying
but
experimentalists can measure some metric of being in a native state.
yeah,
They
right.
can see some deviation
Yeah.
to it, and in some fully denatured state,
Yeah,
they know it has another value.
right.
So you could ask the question, how far along do you get
Yeah.
in that space
Yeah.
before the polymer somehow brings it back?
Right. Yeah, we don't know that, right? So I'll make a couple of quick comments. Like one, I think it's still true that in certain cases that have been studied that it does prevent the denaturation. Like the toluene example, it's not likely helping it refold. I guess I don't know that for certain, but it seems more sensible that it's protecting it. And then there's another case where it's sustained at high temperature and preserves its structure. So that's an indication that for different systems, it can engage in various mechanisms. But I don't know. I mean,
There's any general answer to it.
Yeah, I don't know. And we're hoping, right, that we can do some of the molecular modeling to figure out at least where the polymers would like to go. I'll be honest, when they discovered this thing that it would unfold and potentially start to refold, that was kind of alerted me because I don't think that we're ever going to be able to observe the refolding. Whereas if it was just sort of a static structure, I could do a simulation of an enzyme and see where the polymers are going to come and hang out on the surface, but refolding, no. The other thing I'll say is that whenever I've talked about this in front of somebody who knows quite a lot about biology, say, oh yeah, it's like a chaperone. And so apparently it's sort of anticipated a reasonable in the vein of that sort of physics, this sort of like helping to refold type of thing.
Other question? You have one, go for it.
Oh yeah, you mentioned that you can combine polymer scaling law to do a prediction. I'm curious, in that case, did you incorporate all different scaling law from different architectures like STAR?
Yeah. Yeah, so there's a really there's actually a neat way that for arbitrary topology, you can get the same like random walk style of theory, or actually it's the one thing is that the theory doesn't include any excluded volume, but it turns out for sort of arbitrary topology, you can get the radius of gyration or like the proportionality to that for any sort of topology. Yeah. But it's also looking into that as one reason I didn't get into it, but we're systematically bad for dendrimers. And it's because that theory lacks notion of excluded volume.
Walker?
So, quick question. So, regarding your polymer protein interaction, I understand that if you just isolate the protein, unfolded protein, it knows how to fold itself.
Mhm.
The sequence knows how to fold itself.
Yeah.
So it doesn't stop it from aggregating with other proteins, by
Mhm.
providing some type of a cage for it or something like a chaperone.
Mhm.
It just folds by itself,
Mhm.
right? The sequence knows how to fold. But I don't understand why there was a specificity in your result. That this polymer works only for this protein.
So I'll make two comments, I guess. One is if I understand what you're saying, like if I just want to confirm because I don't know, like if you denature the protein and then you let it hang out at ambient conditions, as long as it's not interacting with other things, it'll refold.
That solution. Find a lot of protein.
Okay. At least on the time scale, I think of the experiments that we don't observe that, but it's not controlled, a single protein, that sort of thing. Our notion of specificity is just that because the enzymes themselves are chemically distinct, right? That the nature of interaction between the polymers and the enzymes is also related to the chemistry. So there's some sort of complementary type of interactions that depend on the balance of hydrophobic versus hydrophilic charge type of residues that is sort of underlying the initial array of monomers that were crafted. And in fact, in sort of the original work from Ting Xu and that sort of thing, they did sort of like a detailed composition analysis, a surface analysis, I think, to what they argue is sort of guide the selection of their compositions that were ultimately deployed. But that required quite a lot of, from my perspective, a lot of work. And I don't know if it exactly worked in that way, but it comes down to that complementary aspect of the polymer chemistry to the enzyme chemistry.
One suggestion, you could also look at really fast folding protein.
Mm.
That you can simulate.
Yeah, okay.
On the interest of keeping on schedule, let's all thank Professor Webb. Awesome talk. Apologize.
No, this is the thing.
But
I bet you there's a really rich conformational diversity.
It looked yesterday.
So there's probably multiple subpopulations